This video is about organization and expression of immunoglobulin genes, models of immunoglobulin gene structure, multi-gene organization of immunoglobulin genes, DNA rearrangements and mechanisms. And this particular study note is developed by EPG Patshala team, Government of India, MHRD. So I would like to express my sincere gratitude to the development team for developing this note. Thank you so much. An animal comes across a large number of pathogens every day that pose a threat to the well-being and sometimes even the life of the organism. The immune system of the animal enables it to survive these encounters with the pathogens. Non-vertebrates possess an innate immune system, however, they do not have much of an adaptive immune system. On the other hand, almost all vertebrates have been shown to possess both innate as well as adaptive immune system. The adaptive immune system protects the animal against a wider variety of ever-changing and ever-evolving pathogens. The characteristic features of adaptive immune system are that it is specific and has immunological memory against antigens. The ability of the animal to launch an immune response against a specific antigen upon re-exposure is crucial for survival of the animal. Recent advances in the field of genomics have shown that immune genes comprising only 7 to 8% of the vertebrate genome, how can then just foster a pattern of antigen specificity arise from the vertebrate genome? This chapter attempts to answer this and other such basic questions. So immunologists realized early on that one of the most important effector molecules for adaptive immunity is the antibody molecule. An antibody is an immunoglobulin protein that is secreted from the B cells and released into the circulatory system. In contrast, an immunoglobulin molecule present on the surface of the B cell is called membrane immunoglobulin. Compared to an antibody molecule, the membrane immunoglobulin molecule has an extra hydrophobic transmembrane sequence. So, an immunoglobulin molecule present on the surface of the B cell is called membrane immunoglobulin. Compared to the antibody molecule, the membrane immunoglobulin molecule is an extra hydrophobic transmembrane sequence. Hydrophobic in the sense water hating transmembrane sequence. When the membrane immunoglobulin recognizes an antigen, its associated specialized membrane proteins like Ig alpha, Ig beta heterodimer help in signal transduction to the interior of the cell. This assembly of membrane immunoglobulin with the associated signal transduction protein is known as B cell receptor or BCR. Each immunoglobulin protein molecule is composed of two heavy and two light chains. The heavy chain of an immunoglobulin molecule is always encoded by immunoglobulin heavy IgH locus, while the light chain may be encoded by either immunoglobulin kappa or Ig kappa K for kappa K or immunoglobulin lambda IgL locus. Further, each light and heavy chain is comprised of a variable region and a constant region. Together, the variable region of heavy chain and light chain determine the antigen specificity of the immunoglobulin. The constant region of the heavy chain, on the other hand, is responsible for the effector functions like complement activation and binding of Fc receptor on macrophages, a process which triggers phagocytosis of the antigen bound to the antibody. Some modes of immunoglobulin gene structure. In the present age of high throughput sequencing and structural analysis, we have a much clearer picture of how the genes are arranged at the immunoglobulin loci and how the sequence variations are brought about. However, in the early days, the absence of sequencing data and the structure information made it particularly difficult for the immunologists to come to a consensus on the issue of immunoglobulin gene structure. Many models were thus proposed which attempted to explain the immunoglobulin gene structure and the origin of immunoglobulin diversity. Some of these are gene line theory. The proponents of this theory advocated that all the variations required for recognizing the foreign antigens were already present within the genome of the organism. They postulated that a large portion of the genome is dedicated to code for the immunoglobulin gene, and this in turn gave rise to a B cell with a vast repertoire of antigen specificities. This theory did not postulate the presence of any kind of mutation or recombination to account for the tremendous amount of antigen specificities. Somatic variation theory. This theory postulated that the vertebrate genome in the germline does not per se encode for all the immunoglobulin genes. Rather, a relatively smaller number of immunoglobulin genes undergo mutation and or recombination to produce the staggering levels of diversity in antigen specificities. In the absence of sequencing data in the early years, this theory was hard to substantiate with evidence. Recent technology has now provided ample evidence for this theory. What is this theory? This theory postulated that the vertebrate genome in the germline does not per se encode for all the immunoglobulin genes. Rather, a relatively small number of immunoglobulin genes undergo mutation or recombination to produce the staggering levels of diversity in antigen specificities. They rearrange or mutate. Then two gene models. Initial sequencing experiments of the immunoglobulin protein revealed that the amino portions of different immunoglobulin proteins tended to be variable while their carboxy portions were found to be more consistent with each other. To account for these findings, Dreyer and Bennett in 1965 hypothesized that each chain of the immunoglobulin chain is encoded by two separate genes, one for the variable region and one for constant region. They further hypothesized that these two genes come together at the DNA level to form a single immunoglobulin protein. However, this theory received a lot of criticism because it was against the first one gene, one polypeptide hypothesis, which was prevalent at the time, and critics found it hard to envisage the possibility that two genes could encode for a single polypeptide. Initial sequencing experiments of the immunoglobulin proteins revealed that the amino portions of different immunoglobulin proteins tended to be variable, while their carboxy portions were found to be more consistent with each other. Then, wait a minute. What is gene model? Initial sequencing experiments of the immunoglobulin proteins revealed that the amino portions of different immunoglobulin proteins tended to be variable, while their carboxy portions were found to be more consistent with each other. To account for these findings, Dreyer and Bennett in 1965 hypothesized that each chain of the immunoglobulin chain is encoded by two separate genes, one for the variable region and one for constant region. They further hypothesized that these two genes come together at the DNA level to form a single immunoglobulin protein. Then, Toneguan's experiment. In 1976, Nobumichi Hozumi and Susumu Toneguana, I'm not sure of the pronunciation, just read the name, Tonegawa, yeah, provided irrefutable evidence for the two gene models by showing that the V genes and C genes join together at the DNA level. In this simple yet elegant experiment, they compared the hybridization profiles of two types of cells after distinguishing them with BAMH1 enzyme. Both cell types were hybridized to radioactive probe derived from either whole kappa chain mRNA corresponding to variable plus constant region genes or from just the 3' portion of kappa chain mRNA corresponding to only the constant region genes. So one of the cell types that were used in the experiment were the embryonic liver cells. These cells contain all the genes in the germline condition, meaning that the genes are the same as they were inherited from the gametes without any somatic alterations. The 3' end kappa probe hybridized with only one DNA fragment of the embryonic cell DNA, while the whole kappa chain probe hybridized with two distinct DNA fragments of the embryonic cell DNA. This means that the germline, the variable and constant region genes are on separate DNA fragments. The other cell types that was used in the experiment was the tumor cells, which express kappa chain of the immunoglobulin molecule. The 3' end kappa probe hybridized with only one DNA fragment of the tumor cell DNA. Further, the whole kappa chain probe also hybridized with the same DNA fragment of the tumor cell DNA. This implied that in the cells which express a kappa chain, the variable and constant region genes are located on the same DNA fragment. So the other cell type that was used in the experiment was the tumor cell, which expressed kappa chain of the immunoglobulin molecule. The 3' and kappa probe hybridized with only one DNA fragment of the tumor cell DNA. Further, the whole kappa chain probe also hybridized with the same DNA fragment of the tumor cell DNA. This implied that in the cell which express a kappa chain, the variable and constant region genes are located on the same DNA fragment. Then, putting together the results of both the cell types, the authors concluded that in the germline condition, the variable and constant region genes are located at some distance from each other, while during lymphocyte differentiation, the variable and constant region genes are joined together at the DNA level to form a continuous structure. Further research in the structure of immunoglobulin genes has revealed that the variable region itself may be encoded by as many as 3 separate genes: variable region gene, diversity gene, and joining gene. That happens in the case of heavy chains. The constant region, on the other hand, is encoded by only the constant C gene. Then multi-gene organization of immunoglobulin genes. Multi-gene immunoglobulin genes make it possible to generate the huge diversity of antigen specificity. The IgH locus encodes for heavy chain, while the IgH and IgL loci encode for the light chain of the immunoglobulin molecule. Most immunoglobulin genes are present as clusters at specific loci on the particular chromosome. However, some immunoglobulin genes are present at loci other than their specific loci too. Such genes are known as orphones. So, since orphones are present outside the loci, they do not contribute to the formation of a functional immunoglobulin molecule. It is noteworthy, however, that not all the genes present at their specific loci are functional either. The non-functional genes are known as pseudogenes. In this chapter, I shall concentrate only on the functional genes at the immunoglobulin loci, as those are the only ones that form a functional immunoglobulin protein. The variable region of immunoglobulin heavy chain is encoded by 3 genes: variable, D diversity, and J joining, while that of light chain is composed of B and J genes only, no diversity. Okay, depending on the haplotype, the IgH locus has 38 to 46, IgK locus has 34 to 38, and IgL locus has 29 to 33 functional V genes. The relative positions of the V, D, J, and C genes in humans, as indicated in the IMGT database, the International Immunogenetics Information System, are illustrated. The presence of multiple VDJ genes is instrumental in generating the variety in antigenic specificity using combinational diversity, which is discussed in the next session. Combinational diversity. As the variable region of the immunoglobulin protein is made up of V, D, and J genes, it stands to reason that if we calculate the total number of possible ways in which these genes can combine with each other, which is known as their combinatorial diversity, we can obtain a fairly good estimate of the possible number of antigen specificities that the human immunoglobulin loci are capable of generating. Due to combinatorial diversity, a total of just 154 immunoglobulin genes have a potential to encode for a staggering 2.26 million different antigenic specificities. Thus, combinatorial diversity of the multiple immunoglobulin genes is one of the most important contributors to the diversity in antigenic specificity. This diversity in antigen specificity is crucial to recognize a wide variety of antigenic determinants found in the pathogen. It is important to note here that the constant genes IgHC, IgKC, and IgLC do not contribute to the combinatorial diversity. Then immunoglobulin locus, IgH, IgK, IgL. It is noteworthy that these combination events are not entirely random events, with some V, D, and J gene segments being used more frequently than the others. However, assuming that these recombination events are random, with recombination events are random with no preference of the one gene segment over the other. It is very practical choice as it makes the calculations of possible diversity very easy. Therefore, in this chapter, I shall consider that for all practical purposes, the recombination events are random. Other mechanisms beside multiple immunoglobulin genes and their combinatorial diversity also play important role in determining the variety of antigenic specificity. These mechanisms are p-nucleotide addition, exonuclease trimming, and N-nucleotide addition, and are discussed in later sections. Difference between somatic and meiotic recombination. DNA rearrangements that occur at the immune loci are important to generate the diversity of our immune system receptors so that our immune system can recognize the vast multitude of potential pathogens in the environment around us. This kind of DNA rearrangement occurs in the lymphocytes, which are somatic cells, and is therefore known as somatic recombination. This is different from meiotic recombination, which also involves DNA rearrangement but occurs during gametogenesis only and does not occur in any somatic cells. So, somatic recombination occurs in somatic cells B and T lineages. Increased diversity in antigenic receptors of an individual. DNA rearrangement is intrachromosomal, occurs between different genes on the same chromosome. Does not involve homologous recombination, involve RAG and TDT enzymes. Then meiotic recombination occurs in germ cells, increased genetic addition between individuals in a population. DNA rearrangement is interchromosomal, occurs between the homologous chromosomes, involve homologous recombination, does not involve RAG and TDT enzymes. Then major events and factors during somatic recombination. Somatic recombination include DNA rearrangement between V, D and J genes at heavy chain locus and between V and J genes at light chain immunoglobulin loci. Further, the T cell receptor loci, TRA and TRB, also undergo somatic recombination. It is of note here that although the germline DNA of all the cells of an organism is the same, the process of somatic recombination of the developing B cells and T cells introduces changes in their DNA, and therefore the DNA of mature B cells and T cells differ from each other and also from the rest of the cells of the body. The rearrangement of immunoglobulin loci is restricted to lymphocytes of the B cell lineage only. Further, even within the B cell lineage, the recombination event is stage specific and immunoglobulin locus specific. IgH locus rearranges first with V to J recombination preceding V to DJ recombination. The rearrangement of IgH locus is followed by that of light chain loci, IgK or IgL in humans. In mouse, the IgK locus rearranges before the IgL locus. Both IgK and IgL do not contain the D gene and thus undergo V to J recombination only. All the lymphocytes are derived from the common lymphoid progenitor CLP. The differentiation of a CLP to a mature B cell is tightly regulated process with many intermediate stages along the way. The first stage after CLP is the pro B cell stage, which is followed by a pro B cell stage. The cells in pro B stage differentiate into cells in the pre B stage and finally into a mature B cell. This process of B cell differentiation occurs in the bone marrow. Once the cells differentiate into mature B cells, they leave the bone marrow and enter the circulation. At this stage, the mature B cell expresses the BCR but has never encountered its specific antigen. Such cells are known as naive B cells. Once these cells encounter an antigen, they become either plasma cell or memory B cell. The plasma cell produce antibodies which are secretory forms of immunoglobulin molecules, heavy chain VJ and VDJ recombination, light chain VJ recombination, V gene, J gene, VJ recombination, recombined VJ. Then in case of this heavy chain DJ and VDJ recombination, V genes, D genes, J genes, DG recombination, VDJ recombination, BJ recombination followed by VDJ recombination at IgH locus, VJ recombination at light chain locus. These genes are many kilobases away from each other. Then stage specificity of events during somatic recombination. Let us now consider in detail how the stage specificity corresponds to the immunoglobulin locus specificity. At the pro B cell stage, the first event to occur is the recombination of one of the J genes with one of the D genes. D to J recombination at the heavy chain locus IgH. In the event of a successful rearrangement at the first allele, the rearrangement at the second allele is inhibited, a phenomenon known as allelic exclusion. However, the process of rearrangement can lead to the out-of-frame joining of the genes, disrupting the entire protein encoding sequence. A codon has 3 bases, and therefore, in 2 out of 3 cases, a rearrangement with out-of-frame joining can occur, which renders the allele non-functional. Fortunately, the cell has a second allele, which then undergoes the D to J recombination at its IGH locus. Those pro-B cells which are unable to undergo a productive D to J recombination on either of the two alleles undergo cell death. On the other hand, a successful DJ recombination event is followed by a V to DJ recombination event. Here we could see this rearrangement stages during B cell development where somatic recombination occurs. CLP, pre-pro-B, pro-B, IGH rearrangement, large pre-B, small pre-B, immature B, mature B. An in-frame V to DJ rearrangement at the DNA test is said to be productive and codes for a peptide which forms the heavy chain of the immunoglobulin protein. However, like VJ recombination, this process too is error prone, and if one allele is unable to undergo a productive V to DJ rearrangement, then the activity is undertaken by the other allele. In the event of a successful rearrangement of first allele, allelic exclusion of the second allele follows. The exact mechanisms of allelic exclusion are under a lot of speculation. However, experimental evidence indicates that the heavy chain peptides formed after a successful V to DJ recombination interact with the surrogate light chain. These chains are expressed on the surface of a large pre-B cells along with Ig alpha, Ig beta heterodimer. The polypeptide assembly thus formed is known as pre-BCR. The pre-BCR leads to intracellular signaling, which ultimately leads to allelic exclusion and cell proliferation. After the rearrangement of IGH locus, the light chain loci undergoes V to J recombination in the pre-B cell stage. Recall that the light chain loci do not have the D gene, and therefore one of the J genes straight away combines with one of the V genes. As in case of IGH locus, if the rearrangement at one allele is not successful, the one allele is used. On the other hand, in case of first allele undergoes a productive rearrangement, allelic exclusion of the other allele ensues. In case of light chains, this phenomenon of exclusion exists between alleles as well as between light chain loci. Rearrangement at one light chain locus suppresses rearrangement at the other light chain locus. This phenomenon of exclusion ensures that only one light chain locus is rearranged and that two at only one allele. Then in case both alleles of light chain locus fail to undergo rearrangement with the correct reading frame, the cell does not die. Instead, rearrangement of the other light chain locus begins again. If the rearrangement of one allele is not successful, the other allele is used, while allelic exclusion of the other allele ensures in case of an in-frame rearrangement of the first allele. The rearrangement IgK or IgL loci encode the kappa or lambda light chains respectively. Either of these light chains replace the surrogate light chain of the pre-BCR. This new kind of receptor formed of two rearranged heavy and two rearranged light chains together with Ig alpha Ig beta heterodimer is known as BCR and is expressed on the surface of mature B cells. Ig alpha and Ig beta are specialized membrane proteins that help in signal transduction to the interior of the cell. All the BCRs expressed on the surface of mature B cell have the same antigen specificity. This is made possible by allelic exclusion because it stops the other allele and another light chain from rearranging its DNA, thus ensuring the presence of only one rearranged heavy chain and one rearranged light chain. Then major proteins involved in somatic recombination. Many proteins are involved in the process of somatic recombination. Some of these are specific for lymphoid lineage, while others, such as those involved in non-homologous engineering or NHEJ, are expressed in virtually all cell types. Proteins encoded by recombination activating gene 1, RAG1, and recombination activating gene 2, RAG2, are a very integral part of the recombination process. These proteins bind to specific sites called recombination signal sequences, RSS, on the V, D, and J genes and help in introducing breaks into the DNA. The expression of RAG genes occur in a stage-specific manner. The stage specificity could, at least in part, explain the stage specificity of the rearrangement of the heavy chain and light chain genes in B cells. High genes of High levels of RAG1 and RAG2 are seen in two stages during lymphocyte development. Pro-B cell stage, where IgH locus rearrangement take place, and small B cell stage, where IgK, IgL loci rearrangement occur. Another lymphoid-specific enzyme is the terminal deoxynucleotide transferase, which is typically expressed only in developing lymphocytes. Recombination signal sequence RSS. Recombination signal sequence or RSS. RSS are DNA sequences consisting of a hectamer, a spacer, and a non-amer sequence. The hectamer sequence is a conserved palindromic sequence, while the non-amer is a conserved AT-rich sequence. Sandwiched between the hectamer and non-amer is a relatively non-conserved spacer sequence, which is either 12 bp or 23 bp in length. Corresponding to the length of their spacer, the RSS are called 12 bp RSS or 23 bp RSS. RSS are found exactly downstream of V genes, exactly upstream of J genes, and both exactly upstream and downstream of D genes. RSS are the sites where the RAG1 and RAG2 proteins bind to the DNA and cleave it. Thus, they ensure that the somatic recombination does not occur at a random sequence, but rather at specific sites only, that is, just upstream or downstream of the gene. This site-specific nature of recombination is the basis of the process of generating a wide variety of antigen specificities. Further proof of the importance of RSS is the fact that recombination at non-specific sites can lead to cancers. So a general rule regarding the RSS is that the 12 bp RSS recombines only with the 23 bp RSS and vice versa. This phenomenon is known as 12 to 23 rule. This rule ensures that the IgH locus J gene combines with the D gene before combining with V gene. On the other hand, owing to the 12 to 23 rule, the V to J recombination always precedes V to DJ recombination. Molecular mechanisms of somatic recombination. Now that we have discussed the major steps involved in somatic recombination, let us now look at the molecular mechanisms underlying this fascinating process. The basic molecular mechanisms underlying the process of V to J recombination are essentially the same as those for V to DJ recombination. Further, the V to J recombination that occurs at the light chain loci also involves the same principles. These somatic recombination processes occur in an ordered sequence, and each, for the sake of convenience, can be broken down into various steps. These steps involved in somatic recombination are RAG binding. The first step in somatic recombination is the binding of RAG1 and RAG2 proteins to the RAS, which are juxtaposed to the immunoglobulin genes. In accordance with the 12 to 23 rule, the RAG proteins bind 112 bp RSS and 123 bp RSS. The two immunoglobulin genes being recombined may be many kilobases away from each other. Then cleavage by RAG. After they bind the appropriate RSS, the RAG proteins produce a single strand break in the DNA between coding sequence and its flanking RSS. This break is introduced at precisely the 5' border of each RSS. And RAG-mediated transesterification. As a result of the single-stranded DNA break, the 3' hydroxyl and 5' phosphate end are generated. Next, the RAG protein catalyzes a transesterification reaction wherein the 3' hydroxyl group at the end of the gene launches a nucleophilic attack on the phosphodiester bond on the opposite non-coding strand. This reaction generates two ends, a coding end having a closed hairpin loop and a blunt signal end. These ends are held together by RAG in post-cleavage synaptic complex. Note that overall two coding ends and two signal ends are generated for each recombination event. The two signal ends generated in the process are joined by DNA ligase IV to form a signal joint. Other accessory proteins, including those involved in NHEJ, are also essential for this process. So, opening of the hairpin and P nucleotide addition. The hairpin loop at the coding and undergoes Artemis mediated cleavage. Depending on where the cleavage occurs within the hairpin loop, a 5' overhang, a blunt end, or a 3' overhang can be generated. Using the strand with the overhang as template, DNA repair enzymes add nucleotides to the complementary strand. This process results in the formation of palindromic sequence and is therefore known as P nucleotide addition. In addition to combinatorial diversity, palindromic nucleotide addition also contributes to diversity in antigenic specificity. In case of light chain V to J rearrangement, the coding sequence of V to J genes after P nucleotide addition are joined by DNA ligase 4 and other proteins involved in NHEG, thus generating a coding joint. Then exonuclease trimming and N-nucleotide addition. Besides P-nucleotide addition, rearrangement at heavy chain genes involve two more mechanisms: exonuclease trimming and N-nucleotide addition. Exonuclease trimming involves cutting of nucleotides at the 3' end. Sometimes trimming can lead to removal of P nucleotides and even the entire D gene. N-nucleotide addition refers to non-templated addition of nucleotides to the 3' end of the coding sequence. This process is catalyzed by the enzyme terminal deoxynucleotide transferase (TDT). After all of these processes are thought through, the coding sequence of the heavy chain genes V and D and J to D to J recombination, V and D4V to DJ recombination are joined together by DNA ligase 4 and other proteins involved in NHEG, thus generating a coding joint. Hairpin cleavage will be happening. Exonuclease trimming for only heavy chain genes, P-nuclease third addition. Palindromic sequence, N-nucleotide addition, ligation by DNA ligase 4 and NHEG proteins, ligation by DNA ligase 4 and NHEG, coding joint. In case of heavy chain rearrangements, the addition or subtraction of nucleotides during pre-nucleotide addition, N-nucleotide addition and exonuclease trimming does not necessarily occur in groups of 3 and therefore can lead to an out-of-frame rearrangement which is unproductive and can lead to cell death. This means the tremendous cost of the organism. However, this process increases the variety of antigenic specificity and therefore are beneficial to the organism. Once the recombination for both heavy chain and light chain loci has been accomplished, the mature B cells start expressing the BCR on their surface. All the BCR on the surface of mature B cell process the same antigen specificity. Then regulation of somatic recombination. The process of somatic recombination is stage specific and follows a particular sequence of events. It is therefore not surprising that it is controlled by a large number of regulatory factors. Some of these factors are cis-acting. They are present on the same chromosome which they are acting. Others are trans-acting. Some of these factors are cis-acting, some are trans-acting. So other kind of factors, the trans-acting factors are not present on the same chromosome on which they are acting and include a lot of proteins. Some of the trans-acting factors are listed. The trans-acting factors are DNA sequences such as enhancers and promoters. These promoters act over a very short range of DNA and are thus present only few bases away from the genes. The enhancers, on the other hand, can act over long ranges of DNA and can thus activate genes which may be many kilobases away. Besides somatic recombination, the immunoglobulin loci undergoes transcription as well. Both these processes are governed by the same cis-acting factors. Each immunoglobulin locus has its own promoter and enhancer sequences. Inappropriate activation or repression of any of these regulatory elements can have a profound impact on the immune system and can lead to many pathological conditions. In conclusion, the process of generation of a vast multitude of antigenic specificities is an extremely costly endeavor for an organism requiring precise coordination between various regulatory factors. Moreover, unprotected DNA rearrangements may lead to cell death. However, given the immense survival value that the process carries for the organism, the benefits of the process far outweigh the cost. Therefore, somatic recombination is fairly conserved during evolution. So now let's have a quick summary. Immunoglobulin molecules are one of the most important effector molecules of the adaptive immune system. An immunoglobulin can be either membrane bound or secreted. The secreted form of the immunoglobulin is known as antibody. An immunoglobulin molecule is composed of two heavy and two light chains. Each immunoglobulin gene locus consists of multiple genes clustered at a particular position on a specific chromosome. The variable region of the heavy chain is encoded by three genes: the variable V gene and the diversity D gene and the joining J gene. The variable region of the light chain is encoded by two genes, the variable gene and the joining J gene. DNA rearrangement occur in a cell type and stage-specific manner. The RAG genes are very important part of somatic recombination. These proteins bind to specific sites called recombination signal sequences on a V, D, and J genes and help in introducing breaks into the DNA. RSS and a DNA sequence consisting of conserved parentomic heptamer, a relatively non-conserved spacer, and a reserved AT-rich nonamer sequence. The diversity of the immune system receptors is brought about by combinatorial diversity of multiple immunoglobulin genes, N-nucleotide addition, P-nucleotide addition, and exonuclease trimming. The process of generation of a vast multitude of antigenic specificity is an extremely costly endeavor for the organism. However, the benefits of the process far outweigh the cost.